Welcome back to Dr. Liu's VCE Biology. Today's topic is the relationship between nucleic acids and proteins, as presented in the 2024 Victorian Certificate of Education, VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. The molecule depicted is a protein, therefore, its monomers are amino acids. In eukaryotic cells, RNA is primarily a single-stranded polymer of nucleotides. Each nucleotide consists of a ribose sugar and one of the nitrogenous bases, adenine, cytosine, guanine, or uracil. A key role of RNA is exemplified by messenger RNA, mRNA, which transmits genetic instructions from DNA to ribosomes for protein synthesis. Gene regulation in prokaryotes controls tryptophan levels via attenuation. This mechanism prevents the completion of transcription, thereby stopping the production of tryptophan synthesizing enzymes. Eukaryotic genes are characterized by a promoter sequence at their start and a terminator sequence at their end, notably lacking the operator region found only in prokaryotic operands. The Golgi apparatus, a complex of vesicles and folded membranes within the cytoplasm of most eukaryotic cells, is involved in secretion and intracellular transport. The primary role of the Golgi apparatus within a cell is to process proteins received from the rough endoplasmic reticulum, modifying and packaging them into vesicles for subsequent secretion, transport, or exocytosis. A comprehensive description of tRNA's function in protein synthesis would encompass its ability to transport specific amino acids to the ribosome for translation. This process relies on the binding of its anticodon to the complementary mRNA codon, thereby allowing for the linkage of amino acids into a polypeptide chain. To explain how an enzyme catalyzes protein breakdown, one could include its ability to speed up the reaction or lower activation energy, the substrate binding to the enzyme's complementary active site, and the subsequent breaking of peptide bonds between amino acids. To describe the role of vesicles in protein export, one could mention transporting proteins to and from the Golgi apparatus, carrying proteins to the plasma membrane, and fusing with the cell membrane to release the proteins. The current model of protein synthesis faces limitations due to its oversimplification, often depicting a linear process that overlooks crucial complexities such as protein folding, gene regulation, and alternative splicing. It also struggles to account for the unknown full genome, a proteome in many organisms, discoveries, and the distinct variations between prokaryotic and eukaryotic synthesis. To overcome these challenges, solutions include employing multiple, more detailed models, pursuing advanced research with new technologies, and adapting existing models in response to emergent scientific understanding. The diverse functions of cells, such as nutrient uptake by intestinal epithelial cells and fat storage by adipocytes, highlight cellular complexity. While these distinct cell types contain the same genes, their specialized roles are explained by their unique proteomes. The remarkable diversity in cell types, exemplified by intestinal epithelial cells and fat cells, can be explained by several mechanisms. Fundamentally, genes code for proteins, and the differential expression of these genes through gene regulation, combined with alternative splicing, ensures that distinct sets of proteins are produced in various cell types. These unique protein sets then fold differently, dictating cellular function. Additionally, Mutations can introduce variations, leading to the production of altered proteins. This section is a key part of your assessment, making up 20 marks, or 16.7%, of the total 120 marks. It includes 4 multiple-choice questions and 2 short-answer questions. The questions are designed to test your understanding of several critical areas. Protein structures. Multiple-choice question 1 will examine the quaternary structure of molecules and their monomers. RNA molecules. Multiple-choice question 2 focuses on identifying the structure and potential role of an RNA molecule in a eukaryotic cell. Gene regulation mechanisms. Multiple-choice questions 3 and 4 will explore gene regulation in both prokaryotic, like E. coli, and eukaryotic systems. Cellular component functions. Short answer question 1 delves into the specific roles of the Golgi apparatus, tRNA, enzyme catalysis in protein breakdown, and vesicles in protein transport and export. Model limitations and cell diversity. Further questions address the limitations of the current protein synthesis model, short answer question 1e, and examine the diversity of cell types in complex multicellular organisms, with an emphasis on gene regulation and alternative splicing, short answer question 2. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.